Thank you for the warm introduction, and thank you for attending my presentation focused on the natural history of the parasitic plant genus Hydnora, which few botanists are familiar with. But at the end of this presentation, I'm sure you'll agree that these are the strangest plants in the world. As you know, the theme of this symposium is plant symbioses. And these particular plants pictured here are plant parasites, meaning that they are plants that parasitize other plants for some or all of their carbon and water needs. Commitment to parasitism can range from facultative to obligate, and hemiparasitism to holoparasitism. I'll illustrate the difference here. On the left, Viscum minimum, a diminutive mistletoe. The small flowers and bracts you see are the plant parasite, parasitizing the larger structure, which is the stem of the euphorbia shrub, the host. Since these floral structures are green, we know that this mistletoe is a hemiparasite. It can produce a portion of its carbon budget via photosynthesis. The odd, fleshy orange flower on the right is the bloom of Hydnora viscerae, the focal genus of this talk. This plant body lacks chlorophyll entirely and depends on its host for all of its carbon and water. Thus, it's considered a holoparasite. The vegetative body of Hydnora are entirely subterranean and lack traditional roots and leaves. These two taxa have had their plastid genome sequenced. Just by size, you can see the plastum of the hemiparasite is typical for a photosynthesizing plant. Whereas Julia Naumann and Stefan Wanka and many other collaborators published the plastum of Hydnora viscerae in the lower right. We showed a highly reduced yet functional plastome that had lost the genes related to photosynthesis. Zooming out, you'll see that parasitism in plants has evolved many times, at least 12 times independently, as you can see on this nice figure drawn by Dan Nickrent. The genus Hydnora, lacking many morphological characters, has been placed variously across the tree of life. Our current understanding places Hydnora either with or within the Aristolochiaceae in the broader Piperales. I like this figure in part because Dr. Nickrent is keeping my study family Hydnoraceae together during this tough year. Julia Naumann, Stefan Wanka, and many other collaborators used a single copy gene approach to place Hydnora within the Piperales, agreeing with a previous study by Dan Nickrent. They also used a molecular clock approach to reveal that Hydnora are among the most ancient parasite lineages, diverging about 100 million years ago. So why are Hydnora species the strangest plants in the world? I know it's a big claim, but I think I can back it up. Just follow along. Just look at the plant on the right, the one that looks like it's sporting orange Shrek ears. That fantastic plant is Hydnora triceps, restricted to a narrow part of the succulent Karoo of South Africa and Namibia. The flower is completely underground, hypogeous, meaning it never emerges from the soil surface. It pushes up towards the soil. Its petals or tepals are fused to form a hood. A few cracks develop in the soil, and then it emits an odor of putrefying meat. The pollinators actually crawl down through the cracks in the soil to the flower to effect pollination. An outline of this presentation today. These strangest plants have an interesting nomenclatural origin story, a fascinating pollination syndrome that I've sort of previewed, a precise mode of germination, and a complex taxonomy. You're somewhat familiar with the flower of Hydnora. Let me introduce you to the subterranean vegetative body, which is anatomically a rhizome. These rhizomes are attached to the host root through a specialized structure in parasitic plants known as a hostorium. You can see this lighter root, which is labeled here, is the host, and it actually terminates at the hostorium. The next arrow points to these lateral appendages or bumps. Each of these have a small meristem that can differentiate into a hostoria or connect to a host root. On to the discovery of Hydnora, an African and Arabian genus. This amazing species was first discovered and described by an apostle of Linnaeus named Carl Thunberg during his travels in South America. This is the first page from his 1775 description of Hydnora africana. Notice the red box here next to Hydnora africana. The issue is that the Swedish term svamp means fungus. This plant was initially described as a fungus. I know what you're thinking. Carl Thunberg is no longer the most famous Swedish Thunberg. So how do we go from fungus to foul flower? Granted, the mistake was bad, but you can see the tooth-like gills of the hedgehog mushroom here really have a similarity with the ornamentation of the tepal margins of many Hydnora species, particularly when dried. Enter Eric Acarius, famous for being Linnaeus's very last student and the father of lichenology. Linnaeus had Acarius redescribe Hydnora as a plant in the genus Aphytia, just 1 year after the original Hydnora description. However, the Hydnorinae, the family name, and thus the genus Hydnora, was conserved in the Montreal Code. Funny sidebar, note the font size of Linnaeus's name versus Acarius's on the thesis cover page. I guess that's what happens when your major advisor is Linnaeus. I really love this Thunberg quote that captures the excitement and mystery of this plant. Just please let me read it to you. But of all that I have so far had the opportunity to see and discover, nothing has seemed to me more strange than this fungus whose description I have the honor of submitting. So strange is its composition that many would certainly doubt the existence of such a plant on the face of the earth. So clearly, Thunberg was on the line in this determination between a fungus or a plant. But clearly, this wondrous plant is a plant. So let's get into the field. This is a coastal area of the Northern Cape province of South Africa, which is also the center of diversity for Hydnora. The green structures here are primarily euphorbia shrubs, which are host to Hydnora species. That tiny white dot there was one of my favorite field sites, just for the absolute isolation and fantastic botanizing. When you're searching for Hydnora, it's honestly tough. You'll often find remnants of dried flowers from previous years, and if your timing is right, you might just see eruptions of the soil that reveal skillfully camouflaged, immature flower buds. But the easiest way to find Hydnora species in the Northern Cape is to follow your nose, detecting the malodorous smell of rotting meat emitted by the flowers. Let's take a look at the floral structure of Hydnora viscerae. With one tepal removed, you can see the chamber flower structure. The fetid odors are produced by a fleshy osmophore recessed into each tepal. An andrecial chamber with a lobe stamen in the form of a ring at the base, and below that, a gynecial chamber with a stigma at the base. Both chambers are superior to the ovary. When you observe the fresh flowers in the field, you see they're often visited by insects attracted to the fetid odor. The beetles crawl on the hairy tepal margin until they reach for the osmophore, and invariably the beetles fall in where they're trapped. They can't crawl out or fly out, and they become dusted in pollen. You'd think that a strong flying insect like a beetle here could just fly out of this flower, but without sort of a minimum takeoff distance, the beetle just can't hover out like a helicopter. In extreme cases, such as this Hydnora esculenta flower in Madagascar, the flowers can be filled with an absolute orgy of beetles scrambling to escape. Close observation has revealed that Hydnora flowers are protogynous, meaning female first. They're in a carpellate stage for several days, followed by the staminate stage, where the antherial ring sheds pollen and the ring slowly restricts. Observations of floral visitors indicated that Dermestes maculatus, a cosmopolitan carrion beetle, was the primary floral visitor. This is the beetle that is used for cleaning skulls and bones in anatomical collections. We also noted that beetles only were selectively trapped, while other insect taxa, roaches and ants, could typically climb in and out of the flower with little difficulty, but beetles were trapped in the chamber flower. Due to happenstance and an unfortunate dead kudu loaded with carrion beetles, I was able to collect enough Dermestes beetles to do an experimental test of the insect trapping mechanism, where I essentially loaded several freshly opened flowers with beetles that I marked with whiteout and then observed their activity for several days. As you can see in this figure, the beetles were imprisoned during the female stage of the flower. Then as soon as pollen was shed, the Dermestes were released by the flower. The pollination syndrome is known as brood site mimicry with insect imprisonment. The actual trap and release mechanism is apparent from left to right. The mechanism itself was a change from a very smooth and slippery floral chamber wall to one that was more textured after pollen release, allowing the beetles to climb right out. And as if this plant could get any stranger, we were able to document with our colleague Roger Seymour that some Hydnora flowers generate heat. For 2 species, we measured temperature and respirometry, and for the last, we measured only temperature. Looking at those results. Thermogenesis results were clear and compelling, as you can see on the figure on the left. For Hydnora esculenta, the nighttime ambient temperature dropped following that blue line from warm to less warm. But you can actually see the pink line indicates that the gynecial chamber actually increases as the ambient temperature drops, a sure case of thermoregulation. Additionally, the figure on the right shows that mass-specific respiration was highest in the osmophore. Again, this is the scent-producing region, suggesting that thermogenesis may be important in volatilizing the fetid odors. Now let's take a quick look at the fruit and seed of Hydnora. As you can see, the fruit is a large berry. It's actually one of the largest fleshy fruits in many of these habitats. Along the Kavango River, you'll find Hydnora abyssinica fruits that are the size of volleyballs. The fruits are edible. I can attest that the pulp is reminiscent of a mealy apple, so just not that great. The Afrikaans names for Hydnora are babianskos or jakkelskos, baboon food or jackal food. It has been long established that Hydnora fruits are relished by animals and humans alike. We were able to collect a number of viable seeds of Hydnora triceps and conduct a germination trial, where essentially we shredded up roots, fresh roots of host and non-host plants and placed them in the center of a petri dish using a technique called the modified cut root method of germination. The results were very clear. The host-specific Hydnora triceps only germinates in response to the root exudates of its exclusive host, Euphorbia dredgiana, demonstrating a sophisticated germination system and probably the formation of a persistent seed bank. Well, how do these hosts get to the roots of their hosts? How do these seeds, rather, get to the roots of their hosts? Well, we found viable seeds in the adorable droppings of this elephant-eared shrew, a lined mouse, and also jackals. These rodents were observed and captured on video, and they spend much of their time at the base of host euphorbia shrubs and burrow there. So we assume they're rather effective seed dispersers for Hydnora. Now, a brief overview of the taxonomy and systematics of the group. Quickly, we generated a 3 loci Sanger sequence data set that resulted in this phylogeny. What stood out most is a clade, the major clades of Hydnora based on host use. The arrow indicates we're a clade of exclusively Euphorbia parasitizing Hydnora species, whereas an earlier diverging lineage parasitized Fabaceae and Commiphora, but primarily Acacia species. You can see that numerical data there. That's stable carbon isotope values of the Hydnora tissues. When they're overlaid on the phylogeny, the CAM photosynthetic signature of Euphorbia clearly demarcate the Euphorbia parasitizing clade versus the clade of C3 photosynthetic hosts, Fabaceae and Commiphora. When floral characters are mapped, other interesting trends emerge. From a fruity floral odor in Hydnora's sister genus, Prosopenki, to a musty gym bag odor in the Fabaceae comifera parasitizing clade, to a rotting meat odor in the Euphorbia parasitizing clade. When endothermy data are overlaid on the phylogeny, there's a trend in a loss of endothermy from the early diverging lineages of Hydnoraceae to the crown taxa, though the data are admittedly scanty with only 4 data points. There are a huge number of Hydnora names to sort through that are reflective of the scramble for Africa colonial phase in Southern and East Africa. Most of these taxa were described by pioneer Italian, German, and English botanists that apparently had little idea of the growing synonymy in the group. But I just like to think that any botanist finding one of these plants in the wild gets really excited and assumes it's something new. The names in green are Fabaceae parasitizing, while the names in red are primarily Euphorbia parasitizing. Over our last 10 years of work, we have simplified the taxonomy, elevated one forgotten species, Hydnora longicollis, described by Velvish from southern Angola, and described one new species, Hydnora viscerae from South Africa and Namibia, and Hydnora arabica from Oman. Many of these synonyms are based on scanty descriptions from East Africa and should be pursued in the field and looking for the type specimens in European herbaria. Some of the challenges of Hydnora taxonomy is a poor condition of specimens. This, for example, is the type specimen of Hydnora esculenta from Madagascar. It's really little more than a shattered slice of Hydnora fruit. Fun anecdote, our description of Hydnora arabica from Oman was aided in no small part to Google image searches, Flickr, and a birding blog that we found online. So what are the factors that contribute to speciation in this genus? Using the Hydnora africana complex as an example, a few drivers of speciation emerge, including summer versus winter rainfall areas, host specificity in the range of those hosts, geographic isolation, and clear differences in phenology, which you can see here, looking at flowering times among these 3 taxa in Namibia. Additionally, in Southern Africa, Hydnora longicollis and Hydnora viscerae follow a well-known species disjunct pattern in the Namib Desert between the northern and central Namib. So what's next? Phylogenies are always a working hypothesis that benefit from more taxa and sequence data. One thing I've really enjoyed during this pandemic is armchair botanizing. I'm sure most of you are familiar with iNaturalist. This is an image of the most recent Hydnora images on iNaturalist. And the photos appear, they're nicely georeferenced and dated. Usually they're good quality. I highly recommend it. A constant stream of excellent photos, really to keep my enthusiasm high for Hydnora. For what I hope you will now agree are among the strangest plants in the world. Thank you so much for listening to this presentation. And I would like to thank my mentors, Erica Moss at University of Namibia and Lytton Musselman at Old Dominion University, among a whole host of other wonderful collaborators and friends and institutions that have helped me along the way, including Ken Wardak at the Smithsonian. Thanks so much, and I'll be happy to address questions during the panel discussion.